Okay, we are going to take a look at um, the next part of this, and that is the Krebs cycle. So far we've had a situation where a case where pyruvic acid has entered into the matrix, losing the carbon dioxide. So stage 2 in the matrix was pyruvate oxidation. Okay, before it enters the Krebs cycle, which is notice it's in the matrix. It's also called the citric acid cycle. So before it enters, it enters as acetyl-CoA. That's pyruvic oxidation, and there's 2 of these molecules for every 1 glucose. Remember, glucose is a 6-carbon compound. It got broken down to 2 3-carbon compounds that lost the carbon dioxide to form acetyl-CoA. Okay, acetyl-CoA. Okay, so these now enter into the Krebs cycle. There's my glucose, there's my pyruvates, 2 of them. They lose a carbon in the form of carbon dioxide. There it is. And they enter as acetyl-CoA. So this is just 1 acetyl-CoA. Now the cycle goes twice for every glucose, okay? And it forms a bit of ATP, NADH, and FADH2. So step 1, acetyl-CoA stokes the furnace. Step 2 and 3, a little bit of ATP, NADH, and carbon dioxide are made. And steps 4 and 5, redox reactions making FADH, okay, and some NADH. Okay, well, this next slide, do not memorize this. I'm only going to go through it once very quickly. 8 steps, okay. Acetyl joins the cycle by combining with oxaloacetate. Okay, that's 4 carbons. That's 2. That's 6. Okay, and these next 7 steps, the oxaloacetate, or the citrate rather, decomposes back into oxaloacetate. Okay, here goes. Isocitrate, a little bit of NADH is made, carbon dioxide, a bit of NADH is made, a bit of ATP is made, FADH, a little bit of NADH. Here it is, the Krebs cycle, or citric acid. So 2 carbons enter, carbon leaves as carbon dioxide, a bit of NADH is made, FADH is made, some ATP, and we double those numbers because there's 2 entering in from 1 glucose, which is C6, right? Watch the animations and these little interactives, they're very useful. Don't memorize any of those steps. Just get an overall understanding of what the citric acid or Krebs cycle does. Okay, this is where the action really takes place. And you've seen this before in photosynthesis. The inner membrane of the mitochondria contains proteins called cytochromes. These are electron carriers. Inner membrane, let's take a look at it. Inner membrane, there it is, contains electron carriers. You've seen this before in photosynthesis. So this is where these processes are very similar. And look, ATP synthase waiting for it. Electrons are going to move through these cytochromes, and oxygen is going to be the final electron acceptor. When hydrogen and oxygen form combined, they make water. Okay, and what's going to happen as we build up protons? Protons are building up in the intermembrane space. They're going to move through the ATP synthase, and they're going to make ATP. Okay, now let's be clear on something here. The electron transport chain does not make ATP. ATP is made as a result of the electron transport chain when protons build up and they move through the cristae membrane, okay, between the intermembrane and the matrix, and they generate ATP through the ATP synthase, okay? So as the protons move through, that's when ATP is made, okay? So electron transport, no, but chemiosmosis, yes. Okay, electron transport chain prevents a large free drop of energy from food to oxygen. Okay, so what this means is that as the electrons are moving through the electron transport chain, they're slowly releasing energy. Okay, so this makes it manageable. High energy, oxidized, reduced, oxidized, reduced. It gets reduced when it gains the electrons, and the final electron acceptor is oxygen. Okay, okay, let's remember this now. We're looking at these little parts of the footprint. The intermembrane foldings called the cristae. There it is right there. This is like bar hopping on White Avenue, okay? There's my White Avenue. I'm gonna go from bar to bar. I mean, you guys are too young for this, of course, but one day maybe. Okay, as the electrons move through, protons get pumped into the intermembrane space. Notice this is like opposite of photosynthesis. Protons are building up in the lumen in photosynthesis, but it's the same thing, same idea, okay? As the protons move through, they can only move back through the ATP synthase, and a whole bunch of ATP is made. Hydrogens are going to combine with the final electron acceptor to make water. So two products, water, ATP. Electrons move through, combine with hydrogens to form water. The buildup of protons in the intermembrane space, that space right here, that is where the protons are going to move through to make ATP. So small amount of energy is released as they pass from protein to protein. Okay, those are the cytochrome proteins. This energy is used to move protons into the intermembrane space. They build up. Oxygen is the final electronic spectrum, very electronegative, so it's got a very strong attraction for electrons. Think of a tug of war. This would be the last person on your team, the strongest person at the anchor. It's the oxygen gets reduced to form water, and a lack of oxygen causes the system to back up all the way to glycolysis because and fat cannot be recycled. I'm going to talk a bit about that a little bit later on. Chemiosmosis, process that produces most of the ATP, 32 to 34 molecules of ATP per glucose are produced through this process, and it uses ATP synthase. Okay, glycolysis, 2 ATP. Pyruvate, none. Krebs cycle 2. Electron transport 0. Chemiosmosis 32 to 34, depending on the cell and that sort of thing. So ultimately, you're going to make about 36 ATP per molecule, glucose. Here we go. 2 ATP, 2 from the citric acid cycle, 32 or 34 from oxidative phosphorylation. Oxidative, why? Because oxygen's involved. Here, these are substrate phosphorylation, these two right here, okay? These two sections here break down the substrate. Here, oxidative phosphorylation. Okay, fermentation, anaerobic respiration. Glycolysis can produce ATP with or without oxygen. In the absence of oxygen, fermentation or anaerobic respiration produce oxygen, ATP. Anaerobic respiration uses an electron transport chain where oxygen is not the final electron acceptor. So some bacteria, for example, use sulfate as the final electron acceptor. Some bacteria are anaerobes. I mean, they don't need oxygen at all. So they'll use things like sulfates instead. Fermentation is where phosphorylation used instead of an electron transport chain. And there's two types of fermentation. There's lactic acid fermentation, alcohol fermentation. Alcohol, pyruvate, is converted to alcohol, releasing carbon dioxide. Alcohol fermentation by yeast is used in making the good stuff here, okay? Notice that's why you get bubbles in your beer, your parents' beer. Okay, lactic acid fermentation. Okay, look at that cheese. Yum. Pyruvate is reduced by NADH to form lactate or lactic acid. Fungi and bacteria for cheese, but human cells also use lactic acid fermentation to generate ATP when oxygen is scarce, and that's in your muscle cells. Okay, so it's eventually converted back to pyruvate in the liver. So here's my glucose. Okay, a little bit of ATP is made, but if there's no oxygen, this pyruvate becomes reduced to form lactic acid. Just a heads up on this. We've been talking about carbohydrates. There's our metabolic pathway for carbohydrates, but proteins also have a way into this metabolic pathway. So do fats. Okay, and fats obviously have a lot more energy. They're going to make a lot more ATP than carbohydrates. And there's our deamination. Remember that got converted into urea and got peed out, and your liver does that. Okay. Okay, poisons. Some poisons interfere with electron transport chain that causes death. Cyanide, hydrogen sulfide. You can see carbon dioxide uh, and cyanide interfere here. They gum up the electron transport chain, and electrons can't move through. That means that no protons build up, no ATP, and this person dies a miserable death. Used in the gas chambers of Auschwitz, for instance. Okay, and that was in a gaseous form. Um, so horrible death. It's also used, say, in rotenone, like insecticide, herbicides, rat poison, I think, as well. Oligomycin. This is an antibiotic. So this is how antibiotics will kill bacteria. Okay, they gum up the ATP synthase. Okay, so light reactions make ATP and ADPH that fuels the Calvin-Benson cycle, which makes glucose. Glucose is used in glycolysis to make a little bit of ATP and a pyruvate. Okay, that can be fermentation, which makes carbon dioxide. We saw that alcohol fermentation. Or if we lose a carbon dioxide, we're going to have acetyl-CoA that enters the Krebs cycle that makes a little bit of ATP, but all those electrons from NADH and FADH fuel the electron transport chain to make 32 to 34 ATPs.